Biotechnology. Table of contents: genetic engineering, diabetes, genetically modified organisms, GMO, gene therapy and stem cell technology. Genetic engineering. Biotechnology is the use and modification of biological systems for the benefit of humanity and has existed for thousands of years. Examples include using microorganisms to make bread and preserve food, and artificial selection to produce more robust animals and plants. Since the 1960s, genetic engineering is what most people think of when biotechnology is mentioned, although it is a subset of it. It is sometimes called recombinant DNA technology or genetic modification. In genetic engineering, the DNA of organisms is modified to alter their functions and traits to deliver new beneficial products, food, medicine, health, etc. This presentation will focus on genetic engineering. Key processes of genetic engineering include DNA sequencing, DNA cutting, electrophoresis, polymerase chain reaction, or PCR, and bacterial transformation and mammalian transfection. The details of these processes don't need to be memorized. It is only necessary to understand what happens and how each process is used. The AP exam could give an example of a technology and ask the student to interpret the given data. The genetic re-engineering process starts by identifying a gene of interest. The target gene can either encode a needed trait that is added to an organism or one that causes a disease that can be removed. Basic research and DNA sequencing are used to identify target genes. DNA sequencing. DNA sequencing discovers the order of nucleotides in a DNA molecule by using multiple laboratory techniques and extensive computing resources. The first step in genetic engineering is to isolate the gene that is to be studied or modified. Computers attached to robotic DNA sequencers fragment, analyze, and find a gene based on user input. DNA cutting. Restriction enzymes, the bacterial defense against viral particles, are used to cut out the gene, producing a DNA fragment. Previous DNA sequencing identifies the location of the gene, enabling the right fragment to be cut out. Restriction enzymes cut the gene of interest from the chromosome by identifying a specific sequence of nucleotides where it will cut. For example, EcoR1 is a restriction enzyme that comes from the bacterial species E. coli. It makes a staggered cut when it reads the sequence GAATTC as shown above. So you see in this DNA sequence, we have GAATTC. When the restriction enzyme EcoR1 gets to this sequence, it recognizes it and makes a cut here between the G and A and on the complementary strand between the G and A. So we get this staggered cut that you see here. Restriction enzymes mixed with DNA is called a digest, because the enzymes break down the fragments of DNA into many smaller pieces. So you can imagine this tube containing many different pieces of DNA, and somewhere in that digest is the gene of interest. Only some of the pieces of the DNA that are in here will contain the DNA that are required for a specific gene. The unique pieces of DNA in the digest must be identified and selected for more analysis. Let's take a look at a gene of interest. The insulin gene between the two EcoR1 cut sites has a unique length. There are DNA fragments of length 5,000, 10,000, and 15,000 nucleotides. The gene of interest is the 5,000 piece. The next step is electrophoresis to isolate the insulin gene. So in the previous slide, we talked about how EcoR1 will make a staggered cut when it encounters a specific gene sequence. So that sequence occurs here and here where a cut is made. What happens on this gene of interest is we get these three pieces. One that has 10,000 nucleotides from this cut site to the end of the fragment. The one in the middle has 5,000 nucleotides, and our gene of interest, our insulin gene, is here. And then our other fragment has 5,000 nucleotides to this end of the fragment. Electrophoresis is used to separate DNA and RNA fragments and protein molecules based on their electric charge and size. There are different types. Gel electrophoresis of DNA fragments will be presented here. The DNA fragments are stained and then placed in an electric field generated by a positive and a negative electrode. DNA has a negative charge, so it is attracted to the positive electrode and moves toward it. Before electrophoresis, it used to be very difficult to find specific genes on DNA. The digest solution that has been dyed, you can see it as this blue color here, is loaded by pipette into a gel that resembles Jell-O. You can see the little drops from the pipette being loaded. The gel is a network of fibers called collagen. Small pieces of DNA can move through the gel quicker than the longer pieces that get tangled in the collagen fibers. This enables the smaller pieces to move farther in the gel. The fragments are separated according to their size and electric charge. DNA has a slightly negative charge. An electrical current is passed through the gel and the DNA fragments move to the positive charge. So you can see up here this start, that was where on the previous slide we could see the digest being loaded onto the gel. And the sample will slowly move toward this positive. Small fragments move faster. Larger fragments are slowed down by the matrix. The result is that the small pieces can travel farther than the larger ones. The DNA is separated by size and electric charge in what is known as a banding pattern. So from our insulin example, we would see three distinct bands, one for the 5,000 fragment, one for the 10,000, and one for the 15,000. The illustration here shows where the DNA is loaded into the gel. Up at the top, slots containing DNA samples, and then moves toward the positive electrode. You can see the arrows showing the movement of the DNA. Shorter segments of DNA go further as shown in the banding pattern. So down here towards the bottom, we would have shorter bands. Up at the top, we would have longer bands. A photograph of the stained DNA separated by size and electric charge is shown. You can see that banding pattern again just in this photograph from the gel. Polymerase chain reaction. Once the gene is located, copies need to be made in the amplification process. PCR, polymerase chain reaction, was designed to make billions of identical copies of a gene within a few hours, making the gene easier to study and to use in genetic engineering. This reaction is carried out by a special machine that utilizes repeating cycles of heat, DNA polymerase, DNA primers, and free nucleotides to build copies of the DNA fragment. Let's take a look at what one cycle of the PCR process looks like. First, DNA is heated to a high temperature, breaking the hydrogen bonds in the DNA, separating the double helix. So you can see in this little like comic star looking thing, we have all kinds of things in our digest, right? We have enzymes, we have the primers, we have the DNA sample itself. And so when we increase the temperature, the bonds that are holding the two black lines here are the DNA fragment. When you heat it, it separates. Okay, so then you get two distinct strands. In step 2, the DNA is cooled and the DNA primers that are in the mixture bind to the DNA segment of interest. The temperature is increased slightly and the DNA polymerase binds to the primers in step 3. The polymerase then attaches nucleotides to the DNA segment. Remember, what we're trying to do here is replicate very quickly these DNA segments over and over and over again. After this first cycle, there are 2 identical DNA double-stranded molecules, which you see up here at step 4. The cycle is repeated, doubling the amount of DNA each cycle, eventually creating billions of copies in just a few hours. Plasmids. To move DNA, vectors such as plasmids or viruses that can be manipulated are needed. Vectors are gene carriers that can move DNA from one organism to another. Natural bacterial plasmids are small, circular pieces of DNA found outside the bacterial genome that reproduce in the bacterial cytoplasm and can move between cells. Plasmids used in the lab are genetically engineered constructs that are used as vectors to move DNA. Engineered plasmids are built in a way that produces, selects, and expresses pieces of DNA. They then move the genes between cells. Plasmids are small extrachromosomal circles of double-stranded DNA found in bacteria. You can see them labeled here, separate from the DNA. Transformation and transfection. Bacterial transformation introduces the selected DNA into bacterial, yeast, or some plant cells. DNA pieces can be recombined to make unique sequences. When new genes are inserted, organisms can express different proteins. The process results in heritable traits. Mammalian transfection is the process of introducing foreign DNA into their cells, resulting in temporary expression or permanent changes in the DNA. This unit will follow the College Board syllabus and just address transformation. Bacterial transformation introduces the selected DNA into bacterial cells. DNA pieces can be recombined to make unique sequences. When new genes are inserted, organisms can express different proteins. This salmon, shown on the left, was given a gene for eel growth hormone, which is more potent than its own and causes it to grow 3 times bigger in half the time. These tomatoes on the right have been given a gene found in fish so they can survive frost, meaning a longer growing season and more yield. Bacterial plasmids are engineered to express inserted pieces of DNA. In addition to a site for the insertion of genes, you can see the inserted gene here, the recombinant plasmids also carry an antibiotic resistance gene so that any bacteria that has a plasmid vector will be able to grow in the presence of antibiotics. an origin of replication so that the plasmids will be replicated in bacterial cells, a promoter so that RNA polymerase will bind to the plasmid and transcribe the inserted gene. Now that the gene of interest is in a plasmid, it can be mixed with bacterial cells and be taken up into the bacterial chromosome. All living things use the universal genetic code. The bacterial cells will read the newly acquired gene, transcribe it to mRNA, and its ribosomes will translate the mRNA into a protein. The bacterial cells will reproduce and express the gene. Each time a recombinant bacterial cell divides by binary fission, it will make a new copy of the gene. The recombinant plasmids are now inserted into bacteria, which will act as vectors. The bacteria cells are briefly shocked chemically, thermically, or electrically, which makes them leaky and more likely to allow DNA to enter. Their membranes recover minutes after the shock. Bacterial cells that pick up new DNA have been transformed. The process is carried out in a test tube with billions of purified plasmids that have our inserted gene and shocked bacterial cells, leading to transformed bacterial cells. At this point, there are now a small percentage of viable transformed bacterial cells. The bacteria are then plated on petri dishes in selection media, a nutrient-containing agar with added antibiotics. Plasmids carry the antibiotic resistance gene, so only bacteria that has been transformed with plasmids could survive and form colonies in the selection media. Only bacterial cells that carry the plasmid will grow on the selection media, since the plasmid carries antibiotic resistance. So we have this agar with antibiotic, and we see these colonies of transformed bacterial cells growing. Bacterial cells without the plasmid will be killed by the antibiotic. The bacterial colonies carrying the genes of interest are expanded by growing them in large liquid cultures. Here's a flow chart that summarizes the genetic engineering process that we've discussed. Start with DNA sequencing with the help of computers. We cut the DNA into fragments using restriction enzymes. We isolate the DNA of interest or the gene of interest using gel electrophoresis. Once we've done that, we can amplify or make many copies of that DNA using polymerase chain reaction or PCR. And then we insert that DNA via bacterial transformation, where we create a plasmid that has our gene inserted, and then we insert that plasmid into a bacteria.